Assalamualaikum and good morning everyone. So today we are going to learn about the organelles, right? Organelles of plant cells and animal cells, since they are very much alike. Okay, when it comes to the content of the organelles. Okay, now let's do some minor recap here. This is a very typical animal cell, all right, which is another example of eukaryotic cell. Okay, so eukaryotic cell, in the previous lesson you learned about the differences between prokaryotic and eukaryotic cell. Okay, and this was the diagram that I show you when I explained about the eukaryotic cell. Okay, and this eukaryotic cell, apparently it represents an animal cell. Okay, so I'm sure you already aware of the organelles that can be found in an animal cell, right? So these are the typical organelles that can be found in an animal cell. Okay, now while this is an example of plant cell, and these are the typical organelles that can be found in a plant cell, the differences with the animal cell is the presence of one, the cell wall, okay, which is, this is the cell wall. You can see the cell wall here, sorry, yeah, this is the cell wall. This is the plasma membrane, which is very similar with the animal cell. And this is the cell wall. You can see how it is being thickened here. Alright, so cell wall, okay, is made of cellulose, right? Mainly cellulose, right? But it has other component like polysaccharides and protein. Okay, but then the main component that make the cell wall is the cellulose. Now, this cell wall is the outer layer that maintains the cell plant's shape and protects the cell plant from mechanical damage. And you notice that there are some holes here, okay, on the cell wall. Now these holes, apparently it serves like a long hollow tube, okay, or a channel. To be more precise, this is a channel, okay, and this channel that is in the cell wall, these channels are known as plasmodesmata. Okay, and this plasmodesmata, right, defined as cytoplasmic channel through cell walls, okay, that connect the cytoplasm of adjacent cells. I mean to say, from one plant cell to the other plant cells, their cytoplasms are connected through this plasmodesmata. Okay, and then we have this chloroplast, which is a photosynthetic organelle. We go into that later. Okay, when it comes to the structure of the organelles. And then plant cell, they have mitochondria, right? And then pericism, very similar with the animal cell, right? The pericism. Okay, and then they also have the Golgi Apparatus. Okay, they have the nucleus, right? With the nuclear membrane, with the nucleolus, right? And things like that. And another organelle that is not present, right, in the animal cell, but only present in the plant cell is the large central vacuole. Some animal cells, they have vacuole, but their vacuole is very small in size. All right, but for plant cell, their vacuole is located centrally and is huge in size. All right, it's a prominent organelle in older plant cells. It's very prominent in a mature plant cell, right, or older plant cell. Okay, in younger plant cell, the size of this vacuole is quite not small, not quite small, but not as big as this one. Right, now there is one structure here, is a tonoplast. Tonoplast is the plasma membrane of the vacuole, right? Tonoplast. And then the cytoskeleton, when compared with the animal cell, right, this plant cell, their cytoskeleton is lack of the intermediate filament, where the cytoskeleton of the plant cell only consists of microfilaments and microtubules. Later we will go into deeper about these microfilaments, microtubules, and intermediate filaments. Okay, so now on comparison for these three cells, the prokaryotic with the animal and plant cells, right? Okay, now the cell wall of the prokaryotic cells usually they are made from peptidoglycan. To say it's a combination of protein with carbohydrates, right? Animal cell, obviously they don't have cell wall, and whereas the plant cell, their cell wall is made from cellulose. Okay, so you can go through on your own. Okay, which one is yes, no, and so on. I just want to discuss on the yellow ones, the ones that's being highlighted here. Okay, now lysosomes. Plant cell do not have lysosome. Please bear that in mind, right? Lysosomes are only available in the animal cell. Okay, chloroplasts only available in the plant cell. Okay, and when it comes to centrioles, centrioles is only available in the animal cell. Okay, centrioles. Okay, now this is how the double helix DNA, okay, they are being folded and compressed to form a chromosome. Okay, now you know that this double helix or DNA double helix, they consist of two strands, right? Okay, now this double helix DNA, to stabilize them, right, what happens, this double helix DNA, okay, will be bound, right, bound to protein compound, and this protein compound is known as the histone, right? Some books mention this protein histone as just histone or histone core, right? So first things first, the double helix DNA will bound to histone protein, okay, to form nucleosome, and then this nucleosome, all right, they are being arranged, okay, being arranged or coiled into solenoids. You can see how this nucleosome being coiled and arranged into solenoids. Okay, and then the solenoid is organized to form a chromatin loop, and then this chromatin loop further, right, further reorganized, okay, to form the rosettes of chromatin loops, and then these rosettes of chromatin loops eventually form the chromosome. Okay, now, so this is the anatomy of a nucleus, which can be found both on the plant cell and animal cell, right? So nucleus, if you look here carefully, okay, is a large structure that's surrounded by a double membrane. You can see the double membrane here. Okay, tengok sini, double membrane, double phospholipid bilayer. Okay, and then a nucleus contains a nucleolus and many chromosomes. Okay, and apparently chromosomes here are in the form of chromatin. Now, the function of nucleus, right? It has DNA, which is being transcribed into RNA synthesis. Sorry, the DNA is being transcribed to synthesis RNA. Okay, and from the RNA will go translation, okay, to form proteins. Okay, or simply the RNA undergoes translation to specify cell proteins. And then we have the nucleolus structure here. It is a granular body, right? A granular body within the nucleus. And then this nucleolus consists of RNA and protein. Now, this nucleolus is the site of the transcription process, okay, or the site for ribosomal RNA synthesis. And this is the nucleolus, also the site where the ribosome subunit is being assembled, okay, which eventually that particular large and small subunit of the ribosome will exit the nucleus and travel into the cell cytoplasm. Now the chromosomes, okay, which is composed of chromatin, okay, is a complex of DNA and protein condensed during cell division, becoming visible as rod-like structure. And then the DNA, the chromosome, right, contains genes, okay, that govern the structure and activity of the cell. Okay, so meaning to say, right, these chromosomes, right, has genes that controls the activity of the cell. How? Because from the genes or the DNA has genes, right? Genes is a small part of the DNA, right, in the DNA. And from these genes, to be more specific, where where this where the RNA can be transcribed. And then when you have the RNA can translate protein, right? Can synthesis protein through translation, and that protein, okay, may control the activity of the cell. Now, next we have endoplasmic reticulum, right? You have this endoplasmic reticulum. They are the extensions from the nuclear membrane, right? So it's a network of internal membranes extending through cytoplasm. Okay. Now, the function of endoplasmic reticulum to synthesize lipids and modify many proteins. Okay, synthesize lipids and modify many proteins. Okay, now when it comes to endoplasmic reticulum, there are two types of endoplasmic reticulum, right? The smooth endoplasmic reticulum and the rough endoplasmic reticulum, right? Dua-dua ni memang berbeza. Okay, the smooth endoplasmic reticulum, as you can see in this diagram, it lacks of ribosome, theta ribosome. Okay, whereas the rough endoplasmic reticulum, it has ribosomes on its surface. When it comes to ribosomes, what's the first thing in your mind? Yes, synthesis of protein. So this rough endoplasmic reticulum manufactures proteins. Okay, while this smooth endoplasmic reticulum synthesis lipids. Synthesis manufactures lebih kurang sama saja. Alright, so meaning to say, we can conclude that the rough endoplasmic reticulum synthesize protein, right? Synthesize and modify protein, right? The protein is being synthesized and modified as well. Whereas the smooth endoplasmic reticulum synthesis lipids. Okay, can you see the difference here? This is the rough endoplasmic reticulum, because you can see there are many black dots which represents the ribosome, whereas this is the smooth endoplasmic reticulum. Okay, next we have Golgi Apparatus. Okay, now this Golgi Apparatus, okay, not usually this Golgi Apparatus is just next to the endoplasmic reticulum, right? So these Golgi apparatus, they are stacks of flattened, you can see how flattened they are, membrane sacs. So these membrane sacs are being flattened, and this Golgi apparatus has two faces. Okay, ada dua muka. The cheese face that facing the nucleus, and the trans face that is facing the cell plasma membrane, right? Cheese face menghadap nucleus, trans face menghadap plasma membrane, right? Now, this Golgi apparatus or Golgi complex, right? They modify, sort, and package protein and lipids, right, from the endoplasmic reticulum into vesicles or transport vesicles, right? And then these vesicles, right, or transport vesicles will be taken by vacuole, okay, or other organelles inside the cell, okay, and as well as the cell secretion. Okay, boleh? All right. So you can see how these lipids, right, from the smooth endoplasmic reticulum, they exited, all right, or exits or leave the smooth endoplasmic reticulum to the Golgi complex or Golgi apparatus. Right, they exit in the form of transport vesicles, not as particles. Right, next, we have lysosome. Right, so lysosome is a membranous sac. Okay, you can see this lysosome is a membranous sac, and within the lysosome, right, there are mitochondrions. Okay, so there is a mitochondrion and peroxisome fragment. Okay, now these lysosomes contains enzymes that break down ingested materials. So I mean to say lysosome can phagocytosize or eat up, all right, eat up materials or food particles. Okay, and then what happens to the ingested materials or food particles? It will be break down, right? Break down into simpler substances, either for the usage of the cell or to be secreted out from the cell. Okay, now we are going to discuss further function of the lysosome. Okay, so lysosome can involve in phagocytosis as well as autophagy. The difference between these two is, right, in phagocytosis, the lysosome, okay, will fuse or phagocytosize, okay, incoming food particles in the form of food vacuole, right, and usually these are from outside the cell. Okay, one simple example is pathogen or any bacteria, right, from the cell that enters, that will be invaginated into the cell. All right, so this lysosome will break down those particular pathogens. Now in autophagy, right, what happens? Any worn out cell, okay, worn out cell or damaged organelle will be break down by the lysosome. Okay, how does this lysosome break down this food or damaged organelle? Do recall that this lysosome has hydrolytic enzyme. So this hydrolytic enzyme will digest those food particles or the worn out organelles. Now, to recap, okay, lysosomes, they are spherical vesicles that contains hydrolases. Those hydrolytic enzymes are also known as hydrolases. Hydrolase, okay, has single lipoprotein layer found in most animal cells and some in immature xylem and sieve tubes. Some plant cells they have, but those are the immature ones, the young ones. Okay, young xylem and young sieve tubes. So when this xylem and sieve tubes matured, they don't have lysosomes at all. Now the functions of lysosomes: digest foreign substances that are endocytosized. Examples are bacteria, viruses, and food particles. They carry out autophagy by digesting all organelles as well as cellular debris. Then carry out autolysis by digesting the whole cell for rebuilding of new tissue during metamorphosis. Example is the digestion of the tadpole tail to build lungs and adult skin. Okay. Now, the endomembrane system. Before I further discuss with you on the other organelles, okay, I want to highlight on the endomembrane system. So this endomembrane system, right, divides the cell so that particular enzymatic reactions are restricted to specific regions within the cell. Okay, maksudnya endomembrane system ni dia khas dia very particular untuk satu bahagian dalam sel tu saja. Okay, now for your level, right, the endomembrane system, right, consists of the ER, the Golgi apparatus, the lysosomes, and the transport vesicles, right? So Pericisms and the vacuoles of cells, they do not communicate with other organelles in the endomembrane system. So these pericisms, okay, and vacuoles, they are not part of the endomembrane system. Only the ER, Golgi apparatus, lysosomes, and transport vesicles are in the endomembrane system. Now, class, you might be wondering, all right, what about the ribosomes in the cytoplasm? Those ribosomes in the cell cytoplasms synthesize proteins, very much similar with the ribosomes on the rough endoplasmic reticulum. It is just that the protein synthesized by the ribosomes in the cytoplasm, or cytoplasmic ribosomes, these proteins are being used in the cytoplasm. Okay, so meaning to say it is being used within the cell. All right, the protein that is being synthesized by the rough endoplasmic reticulum, usually it will be secreted out of the cell as the cell secretion. So now, while in the endoplasmic reticulum, the proteins are folded and form their final 3D shape. Okay, as you can see from here, okay, proteins, okay, from the rough ER are being transported to the Golgi apparatus, where this proteins is being modified and packaged right into transport vesicles, and this transport vesicle will exit the cell as cell secretion. Okay, the same goes for the lipid, which is being synthesized by the smooth endoplasmic reticulum here. Okay, so this lipid will be transported into the Golgi apparatus for modification and packaging, right? And these lipids, either it can exit the cells as cell secretion, or okay, it can be used by the lysosome. Okay, now other organelles, all right, we have the vacuole, right? So vacuoles, they are all right. Now vacuoles, okay, they are membranous sac, right, and usually can be found in plants, fungi, and algae. Then you recall chloroplast is the plasma membrane of the vacuole, right? So vacuole store materials, waste water, okay, and maintain the hydrostatic pressure inside the plant cell. Next, we have Pericism, right? So it's a membranous sac that contains many enzymes, many types of enzymes, all right? So this Pericism, they are site of many metabolic reactions, okay? And one of the main function of Pericism is break down fatty acids. Next, we have mitochondrion, right? Or a mitochondria. Okay, so a mitochondria is a sac, okay, that has two plasma membrane. You can see from here. This is the outer plasma membrane or outer membrane, and this is the inner membrane, right? This is the inner membrane. Now this inner membrane, right, somehow some part of it folded, all right, it folded inside. You can see here how this inner membrane folded inside. Now the part of the inner membrane that folded, all right, is being folded inside or towards inside, this part or structure is known as the crista or cristae. Okay, crista or cristae. This is not the crista. This is the inner membrane of the mitochondria. This is the inner membrane of the mitochondria. All right. This point all the way here and here, this is the crista. The same goes here. This is the crista, 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 all right, crista, and crista. Yang di luar ni semuanya inner membrane, right? Don't confuse and get your fact right. Right. Now between the inner membrane and the outer membrane, okay, there is intermembrane space. Apakah intermembrane space ini? Okay. And then di dalam mitochondria itu we have the DNA, right? Or you can say in the matrix of the mitochondria, okay, we have ribosomes and DNA, right? This is the matrix of the ribosome. We don't call it cytoplasm of the ribosome. We call it matrix of ribosomes. Matrix of mitochondria, right? So mitochondria, right, is the site of most reactions of cellular respiration. Later in the cellular respiration chapter, you will learn a big deal about this mitochondria, right? How it plays a role, big role. So the main function of mitochondria is to synthesize ATP, adenosine triophosphates. Okay. Now next we have chloroplast. Chloroplast is one of the example of plastids, right? So chloroplast is a double membrane structure, right? You can see here it's a double membrane structure enclosing internal thylakoid membrane. Okay, now please take note, this is the thylakoid membrane, right? Where on the surface of this thylakoid membrane there are many chlorophyll. We have chlorophyll A, chlorophyll B, right? Okay, and we also have the carotene pigments. Okay, so those chlorophyll A, chlorophyll B, those carotenes, xanthophyll, phyophytin, they are all on the surface of this thylakoid membrane. All right, and then this thylakoid membrane, they are being stacked. All right, you can see these thylakoids. So, okay, let me rephrase that. This is a thylakoid membrane, right? So this thylakoid membrane, okay, it encloses the thylakoid. So this is one thylakoid, right? So this is one thylakoid. Ini baru tidak clear. This is one thylakoid, and on the surface of this thylakoid membrane is where we have the chlorophyll, right? Okay, or chloroplast. Now these thylakoids, when they are being stacked, okay, these thylakoids are known as granum, okay, or grana. Okay, so granum consists of stacks of thylakoids, right? And when it comes to thylakoids, on the thylakoid membrane we have the chloroplast. Okay, now the matrix or cytoplasm of this chloroplast is known as stroma, and in the stroma there are many ribosomes and DNA. Okay, now look closely on the membrane or plasma membrane of this chloroplast. Okay, we have the outer membrane and we have the inner membrane. Ini inner membrane, ini outer membrane. Okay, kepala saya mix up ni. Alright, now class, again, before I move out, I move into the other organelles. Alright, now this is a chloroplast. In a chloroplast, there are many thylakoids. You can see these thylakoids. Right, and stacks of these thylakoids are known as granum or grana, right? And then on the surface or plasma membrane of this thylakoid there are many chlorophyll pigments. Okay, there are many chlorophyll pigments, and one chloroplast has many granum or many thylakoids. Okay, so functions, okay, the chloroplast, okay, chloroplasts are the site for photosynthesis. Why? Because in one chloroplast, you have this thylakoid where on the surface of the thylakoid membrane there are many chlorophyll, and this chlorophyll, chlorophyll captures light energy, right, for photosynthesis. So the function of chloroplast is to drive photosynthesis or to enable photosynthesis. Okay, next we have the cytoskeleton, right, which is the cytoplasm of the cells. Now, when it comes to cytoskeleton, when it comes to the cytoplasm of the cells, mainly it is formed from the cytoskeleton. And when it comes to cytoskeletons, there are three components. We have the microtubules, we have the microfilaments. This is the microfilaments. Where's my cursor? Right, microfilaments, and this is the intermediate filament. Now, the microtubules, as you can see from here, they are hollow tubes made from subunits of tubulin protein. This is the alpha tubulin, this is the beta tubulin. Right. Now, the function of this microtubule is to provide structural supports, to the cell. So, these microtubules have role in cell and organelle movement as well as cell division. And these microtubules also is a component of the cilia, flagella, centrioles, and basal body. So, meaning to say, cilia, flagella, centriole, and basal bodies, they have these microtubules. Next, the microfilaments, or also known as the actin filaments, is a solid, rod-like structure consisting of actin protein or actin filaments. So these microfilaments or actin filaments provide structural support, very similar with the microtubules. And apparently these actin filaments play a role in cell and organelle movement as well as cell division. All right, next we have the intermediate filament, which are tough fibers that are made of protein. You can see this fiber, structure of the intermediate filament. All right, so this intermediate filament, it helps to strengthen the cytoskeleton, which in turn stabilize the shape of the cell. Next we have the centrioles. Is a pair of hollow cylinder. You can see the centrioles here. A pair of hollow cylinder is located near the nucleus. And each centriole consists of 9 microtubule triplets. Boleh nampak kan sini? Ada 9 triplet microtubule. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's why it is also the centriole. The centriole also, when we discuss about their structure, they have 9 times 3 structure. Alright, 9 times 3 structure. Alright, now this centriole, it is important during the cell division, as a pair of this centriole forms the centrosome. During the cell division. Now if you recall, during cell division, especially mitotic or mitosis process, what happens? This centriole will form the spindle fiber, between one end to one end of the cell, in the cell. So mitotic spindle fiber forms between centrioles during animal cell division. And then this centriole also may anchor and organize microtubule formation in animal cell. Now cilia. Animal cells, they have cilia. So cilia, they are short projections extending from the surface of the cell. So this is the cilia, basically. So the cilia consists of microtubules, and this is the basal body. Basal body means the part of the plasma membrane where the cilia are being projected out, right, or extended. Okay, now cilia, okay, made of two central and nine pairs of peripheral microtubules. So when it comes to their structure, okay, cilia has 9 plus 2 structure. Okay, now flagella. Flagella is the tail of the cell. All right. Prokaryotic cells, they have flagella, right? Most of them. But for eukaryotic, some eukaryotic cells, they have flagella. One example is the sperm cell, right? So flagella, okay, is the long projections that made of two central and nine pairs of peripheral microtubules. So flagella has 9 plus 2 structure as well. Okay, and it extends from the surface of cell and covered by plasma membrane. So both cilia and flagella is not shown here, okay, both structure, okay, cilia and flagella, they are covered with plasma membrane. All right, so cilia is very important in the movement of some unicellular organism, like for example the Pericism. They have many cilia, right? So those cilia enables this Pericism to move from one point to other point, from one location to another location. Okay, and cilia also important in cell signaling, right? Apparently. Now for flagella, not shown here again, is very important in the locomotion of the cell, right? As well as some unicellular organisms. For example, is this one unicellular microorganisms. Apa nama dia? It is under the flagellates. Flagellates. I can't recall the example. I know it's under flagellates. Alright, flagellates. Tak ingat. Okay, whatever it is. Alright, flagella. Okay, it is important for cell locomotion. Now, for animal cell, for the easy example is the sperm cells. So that's it for today. Alright, so see you in the Google Meet later on. Happy studying. Bye.